Okay. So my name is Denis, and I work in Denmark Technical University in Novo Nordisk Foundation for Biosustainability. It's a fancy word for basically biotechnology. And I will tell you about approximate Bayesian inference in the complex industrial scale models. Okay, so a few words about biotechnology. You can meet it everywhere. So food is biotech, drugs are biotech, and now we are slowly moving toward chemical production using biotechnologies, and this is actually the main focus of our center. So how we actually do it? We have those small, really cute bacteria or yeasts, and they are living in very huge tanks called bioreactors. And when we feed it with sugar or some, for example, municipal waste, they happen to produce something useful, say plastics or aforementioned fuels, drugs. Say, for example, we feed them with glucose and they produce part of spandex. How it actually happens inside the cell? So, inside the cell, there are floating around the molecules, and those molecules can sometimes convert into each other, and this property is defined by the thermodynamics. It's very established framework of thinking developed by physicists, but also biologists discovered that this process is also governed inside the cell by the huge protein molecules called enzymes. Those are kind of like machines, and when we have a reaction converting molecule substrate to molecular product, we have like amount of molecules converting per time. So you see here only one error, but in fact we have lots of such reactions inside the organism. How we actually make those cells to produce something? Well, we have an iterative way of doing it. We start from a genetic design. We have a set of genetical designs which we hope will work. Then we try to implement them using molecular biology tools. After that, we test those molecules and strains in the analytical chemistry or workspace. And after that, we get lots of data. But the making sense of it is actually very hard, because biology is very nonlinear in its nature. We have only partial knowledge of our systems, and we have pretty few samples. Usually, most of them are replicates, which make an application of the classical machine learning algorithms to be quite hard. But we want to learn something, and we want to guide our design tools better. So what we can do and what we actually have. We have so-called omics technologies. You probably know about genomics. It's a tool which can tell us what are the genes inside the organism. So we can use the genome information to decipher what kind of reactions we have inside the cell. But now we have also information about other parts of cells, say the small molecules that are floating around, those protein machines that are inside the cell, and now we also can have an estimate of how busy are those reactions. However, biological data is noisy, and we also don't have an exact clue of what is the structure of that noise. Because the variables can be also very correlated, and we don't have a very exact point of view on how to model it. So, what to do? First of all, we need to describe metabolism with some kind of mathematical framework. This is part of metabolic network. It means that this chemical is converted to this chemical, and this chemical can be converted to those chemicals. It's very closely resembles the transportation problem. Say you are trying to go from here to here, and here's the we can express in matrix form how you can actually move through those metabolic network. And those actual conversions, they can be expressed as system of differential equations. They are describing how the concentrations of like of some molecules change over time. What we also now have during like the 100 of years of research is now we have a model of how the actual conversion happens during time. This called Monod-Wieman-Chanin model. It's around like for 50 years at least, and it's very complex. But what's really nice for us is that most of the parameters we first can make some sense from it, and from some of them we can just directly measure using those omics tools, and other parameters we can like easily sample if we use correct reparameterization, or we just want to actually feed them. And what is like why we actually need these models, because they have a predictive power. So this model can predict amount of metabolites inside the cell, or like amount of enzymes and so on, which are very like, and basically they are crucial functions for us. So we're very like easy ABC reminder. Umberto done a great job describing all of this framework. So we actually have a very complicated simulator model, and we have very few experimental data points, because experiments in such kind of biology have a tremendous cost. So here's what we come up with. This one is a very like kind of toy model for the metabolic network. It's like easy. It's like comparatively easy. It is like describing very well-known system inside the hepatocyte. It's a human cell in the liver, and like to not go into details, it can be expressed using 5 ODEs. One algebraic equation is just basically like the balance equation, and it consists like of 72 parameters using all of those MVC models. What is actually very important is that we have a published, not us, the different groups have published models which are very extensively characterized, and we can use the prediction of those models as a training set. So our framework called ABC Grasp, inputs are basically everything that we can collect. It's a data from all the omics tools, the data about the structure of metabolic network, so basically the kind of reactions that happen inside the cell. And we also need some information about how those protein machines are operating. Like this is basically task for biochemists, and for most of the machines it's pretty well known. So how our algorithm operates on the very high level is just first it can be magical. We can't feed a very complicated model with only a few data samples. But if we have a way to create a prior very smartly, we can use that to actually have a very good predictions. So we have a way to create the prior in such way that it will be feasible in sense of like thermodynamics and the general physics, and like the common sense. Say for example, the ratios of enzymes should sum up to 1, because like they can be like it's 100% in some, and other kinds of rules. And after that we have a set of those parameters that are actually like pretty sensible and good. We use those parameters to simulate our models and then compare it to a very few data points. Remember that Umberto said that it scales pretty harsh with a number of points, so we have like no data, no problems, right? So to guide you through all of that, so it's basically like how we can construct that prior. It's basically all we know about biology is condensed to like a small set of rules. Then we use that kind of good parameters to estimate how our system will behave. The simulation is basically feeding those parameters into the ODE model and then finding steady state point. Then we compare that steady state point with the experimental data, and if it's fine, then we just keep those parameters. So it's basically that rejection sampler that was discussed before. And then we are like slowly crawling, moving to the sequential Monte Carlo based sampling. But it's pretty like simple scheme. The epsilon here is set to 2. It's like a balance between speed and accuracy of our results. So, what kind of training data we use? We have those published model, which we can use to generate some kind of experimental inputs. And what's interesting that only some of those generated points exhibit some kind of like difference in in somewhat well-known data. We have kind of like a special reference, which means kind of like the zero state of our model. It can be created from the literature, or we can just set one experiment by ourselves, which will stay like the regular common organism. Those perturbations are made with respect to this zero zero individual. Say, for example, the first perturbation is like it's too hard. The third, for example, perturbation is a knockout of some gene, which leads to loss of some protein machine. And you can see that there are like a couple of blue squares. It means that the data is reliably different in this perturbated model compared to our reference model. So, what kind of results we have? So, this is distance from the prior, and this is like number of data sets. It's basically the iterations of our training. So this one is only one data point that is fed to the model, and here we see like the second data point. Our prior, like our new like posterior, moved completely from the prior, and after that, this distance is basically the same. And also we can see that structure here, it also means more or less the same. So the posterior moved away from the prior, and you can see how it looks like. So this is prior. It's just like you don't need to understand completely what those values are. It's some kind of special properties of that metabolic network that we can metabolic network linked predictions. They are described here. It's like we don't have any data here, right? It's something like that. We have only 2 data points. You start to see some kind of squares, and here's the the posterior prediction after 12 data sets. But you can see that the true control structure that we know from that published model is basically the same. Even from the 2 data points, we start to recover the original biology of that model. And we don't have a very mathematically rigorous approach, but we actually need something that works. Here we can see how we are doing the parameter estimation. Well, the parameter estimation is pretty bad. It's even notice that it's a log scale, so it's like ±1 kilometer. But the actual predictions, surprisingly, are good, because you can see them. They are almost all the times are under the red dots, and even if we don't have a very accurate predictions, it's something that we can explore better afterwards with a continuous experimentation. And after each experiment, we will refine our posterior and make it even better. So we can have a iterative process of improving our posterior and making better and better with every iteration. I think it's one of the most important things for us in industry, because it's not only about one set of predictions, it's about the continuous process of improvement. So it's kind of like an active learning paradigm. And also a very funny test for our model is from the biochemistry we know pretty well about what kind of reactions are there, but we don't know what kind of modulators of that affections are present in the cell. So from the kind of like ground truth model, we remove some of those interactions. And we now think, like, for some reason that, okay, we probably missed something. It's like kind of like biological question. We are interested in uncovering those kind of hidden regulations. So what we are trying to do, then we just like try to sample all the possible kind of regulations. So we just like try to connect every dot with each other, one by one. And then we can simulate those models with the added interactions and try to use Bayesian factor as a way to kind of like measure which model is the best. So what kind of added interactions is probably the best at explaining our data. And looks like that this interaction, which was like which was deleted before, it was recovered, and it was one of the best models here. Like Bayesian factor is more than 3, but like actually it's 20. So it's like, well, we can dance. And all models, they have like not even a consolatory prize. Like, screw them. And like, in fact, we tried to do this for this one, for this reaction, but we don't have any good model explaining that. We don't need it. It probably represents the lack of ground truth knowledge in biology. We have only like kind of assumptions about that. So it can probably be a very great tool to actually do like a data-driven analysis of the biology. So we have actually quite some challenges. So computation load is quite big, even for the toy model. It's around 24 hours to sample that. Well, MATLAB's environment is quite hard to use, especially in computational biology environment. Most of the people and also like myself and all the center is using Python. So we need to probably ditch MATLAB and go to something else. It's actually quite hard to control the diversity of the samples. Effective sample size can be pretty bad if you use some non-very sophisticated sampling algorithm. Also, it's quite complicated to share and communicate resulting model because it's kind of like a bunch of samples. What can we do with that? Like publish it on GitHub and like link it? We don't know. How to scale this solution to higher dimensions? It's kind of like an inherent problem of molecular Monte Carlo. And our prior is very complicated in the sense it involves like several linear programming routines, and we have like lots of hyperparameters, and this is like one of the core parts of the program itself. So moving forward, we have some idea of how we can improve that. We use better Monte Carlo methods, probably switch to variational bias methods because they also propose variational bias methods with intractable likelihood. Probably move to some better numerical foundations, say like TensorFlow probability or Pyro. But we're also very happy to hear your suggestions. Probably you know something. So, going to conclusions, it's really good to use prior knowledge and not to just rush to big data solutions when you just like fit in the neural network with something. We can also use very complex and complicated models within ABC framework. We are mostly interested in prediction accuracy, and we that nature does not care about the numbers. The parameters are like highly, they are not very important for us. Surprisingly, not all data points are equal. We can learn something from the 2 data points, but you have seen that the 10 data points afterwards, they were not so they were not so informative. And in fact, it's still pretty tricky to set up and perform ABC for the complex models. But there is a lot of progress in the field. So thank you. If you have any questions, I would gladly ask. I answer. Sorry.